Hello? 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 Can you hear me? Hello? Can you hear me? Okay, thanks a lot for reply. Can you start? Okay, thanks a lot. Can start. Now, my name is Rohit Shankar Mane, and today we are going to enter in the unit Applied Biology. Today we are going to enter in unit Applied Biology with chapter first, that is a microbial fermentation. Can you see my slides? Okay, we start with microbial fermentation. Now the first question arises when we hear the applied biology and the microbial fermentation. Now first question is what is applied biology? Now applied biology is defined as the combination of theoretical and practical knowledge of the biology for society use. What is applied biology? So applied biology is nothing but the combination of theoretical and practical knowledge of biology for society use. In this unit, we are going to discuss about first chapter, that is microbial fermentation. Now, what is microbial fermentation, how it is carried out, and what are its uses, that we are going to see in further presentation. For the same PPT, I revealed a 10 years back NET, SET, GATE and Indian Institute of Science Bangalore question papers, and I come to know that many questions comes from below chapter. Now the microorganisms and raw materials used in the fermentation procedure, products from fermentation, conditions used in fermentation, recovery process of products, design of fermenter, types of fermentation procedures, and the last one, effect of different environmental factors on the fermentation procedures. Now these are the main points on which many questions came on in NET exam and GATE and SET exams also. By taking this all background, I designed this PPT and implanted below topics. In that, first introduction, followed by biological role, biochemical role, industrial fermentation, design of fermenter, types of fermenter, and the microbial growth during the fermentation procedure. And in the fermentation procedures, we are going to talk on different products like alcoholic fermentation, lactic acid fermentation, mixed acid fermentation, antibiotics production, amino acid production, enzymes production, vitamins production, vaccine production. These are the some important products which I chosen because on these topics many times questions arise in NET, SET, GATE exams. That is why these topics are included. In this presentation, what I am going to do is, first I am going to read first slide, then afterwards I am going to discuss this slide with you people, and at last we are going to discuss questions from that slide. Now here we go. The first one is introduction. Now fermentation is a metabolic process that consumes sugar in the absence of oxygen. The products are organic acid, gases, or alcohol. It occurs in yeast and bacteria in oxygen-starved muscle cells, as in the case of lactic acid fermentation. The science of fermentation is known as zymology. In microorganisms, fermentation is the primary means of producing ATP, while degradation of organic nutrients anaerobically. Humans have used fermentation products, foodstuffs, and beverages since the Neolithic age. For example, fermentation is used for preservation in a process that produces lactic acid in sour foods as pickled cucumbers, kimchi, and yogurt, as well as for producing alcoholic beverages such as wine and beer. Fermentation occurs within the gastrointestinal tracts of all animals, including humans. Now see, now we know that fermentation is a metabolic process that consumes sugar in the absence of oxygen. Meaning is that fermentation is an anaerobic process. Now, according to the Louis Pasteur, what he told that fermentation is an microbial process rather than biochemical or chemical. So according to Louis Pasteur, fermentation is a metabolic process. It is an anaerobic process where the microorganisms are involved. Now majorly yeast cells are involved, for example Saccharomyces cerevisiae. That's what the fermentation products are majorly fermented in type. And the fermentation study is known as zymology. You have to remember this mostly, that fermentation study is known as zymology. Now, as far as fermentation is considered, fermentation is used for the production of different types of compounds such as organic acids, amino acids, antibiotics, etc. While in another way, fermentation is also used for the preservation of foodstuff. For example, pickled cucumbers, kimchi, and yogurt is there. Now, fermentation occurs within the gastrointestinal tract of animals, including humans also. Now, majorly the main points from the first slide is fermentation is a metabolic process. Fermentation is an anaerobic process because it occurs in the absence of oxygen, and the products are organic acid, gases, and alcohol. It is an microbial process. The study of fermentation is known as zymology, and the fermentation use is for the preservation of food products, and it is also used for the production of different types of products, industrial important products. Now these are the questions, very simple questions selected from NET GATE set question papers. Now first question is, fermentation is dash process. So simple answer is microbial process. Fermentation is dash process. So aerobic, anaerobic, both, none of the above. Yeah, it's a B. Now second, fermentation is dash process. So it's a B, that is anaerobic process. Now tell me, study of fermentation is known as what? First A, B, yeah, option B. It's the correct option. Yeah, option B. Study of fermentation is known as zymology. Now fourth one, fermentation occurs in of humans and animals. Yeah, yes. This is also fourth one. It's a D. Yeah, Venkateshwaran. Right, good answer. This is the B. So these are the simple questions which was from a first slide. Now we are going to move second slide, that is biological role. Now see, along with photosynthesis and aerobic respiration, fermentation is an extracting energy from molecules, but it is the only one common way prokaryotes and eukaryotes. It is therefore considered the oldest metabolic pathway for an environment that did not yet have oxygen. Now, yeast, a form of fungus that we know, yeast is a fungus. For example, Saccharomyces cerevisiae is famous yeast cell that we use in different types of fermentation products. Now, yeast, a form of fungus, occurs in almost any environment capable of supplying microbes microbes from the skin of fruits and to the guts of insects and mammals and the ocean, and they harvest sugar rich materials to produce ethanol and carbon dioxide. The basic mechanism for fermentation remains present in all cells of higher organisms. Mammalian muscle carries out the fermentation that occurs during periods of intense exercise where oxygen supply becomes limited, resulting in the creation of lactic acid. Now see, from this slide we can learn the biological role of fermentation. Now we know that fermentation is an anaerobic process, while we also know that there are there is aerobic respiration. Now from the aerobic respiration we can get the ATPs, meaning is the energy. So fermentation is a process where extraction of energy occurs. You have to remember this sentence, that extraction of energy occurs from molecules. Now for that purpose we are using different types of microorganisms. The famous example is the yeast cell, known as majorly known as Saccharomyces cerevisiae. And now the yeast cell is used in different types of products like ethanol fermentation, methanol fermentation, etc. Now this is the yeast cell. The first figure is yeast cell that you can see. That yeast cell is majorly used in the different types of dairy products such as bread, yogurt, etc. And second figure is the yeast cell structure. If we start from inside, then inside we can see the vacuole. After vacuole there is a mitochondrion. Yeah, you can see. Then after mitochondrion there is a nucleus. Then there is a cytoplasm, cell membrane, and cell wall. Now outermost covering is the cell wall that we know. After cell wall there is a cell membrane. There is what? Cell membrane. There is a cell membrane and nucleus and cytoplasm. Now in this you have to remember what you have to remember, that the cell wall of yeast cell is made of from chitin. This you have to remember. In invertebrates, fermentation also produces succinate and alanine. Fermentative bacteria play an essential role in the production of methane gas also, which ranging from the ruminants of cattle to sewage digesters and freshwater sediments. They produce hydrogen, carbon dioxide, formate and acetate and carboxylic acids. And then consortia of microbes convert the carbon dioxide and acetate to methane energy. Acetogenic bacteria oxidize the acids, obtaining more acetate and either hydrogen or formate. Finally, methanogens convert acetate to methane in the presence of archibacteria. Now we know that methane production majorly be known as biogas production. Now in the biogas production or in the methane production, what happens actually, there are different types of microorganisms. We use the methanogens for the production of methane. Now the archibacteria are also used in the production of methane, who carries the conversion of acetate to methane. Now the questions. Basics of fermentation. This question is from IIT Dharwad, which was for PET exam, PhD exam. Basics of fermentation is known as what? Extracting energy, B, invest energy, C, both, D, none of the above. Option C. Option C. Okay, wait. No, option C is not correct actually. Extracting energy. Option A is correct because we are extracting the energy from the raw material by using the microbes. So, Akanksha, very good answer. Now, second is the oxygen supply is limited in humans means what? Need of energy. Second, need of fermentation. C, lactic acid production. And D1, all of the above. What is the need when oxygen supply is limited in humans? There is a need of energy, or there is a need of fermentation, or there is need of lactic acid production or fermentation. And D1, none of all of the above. Akanksha, A. No, A is wrong answer. Anyone? What is the correct answer? Oxygen supply. This question is from Indian Institute of Science, Bangalore. What is the correct answer? Venkateshwaran is saying C, lactic acid production. Yeah, Akanksha is saying D. Yeah, D. It's a very good answer because see, oxygen supply is limited in human beings. Means what? There is a need of energy. Okay. Then next one, need of fermentation, lactic acid production. If we want to do lactic acid production, means there is a fermentation. After fermentation, the lactic acid production will happen or further products. And if the fermentation is occurring, means need of energy. So all factors are considered. So that's why oxygen supply is limited in human beings means what? Need of energy, need of fermentation, lactic acid production, and the answer is so all of the above. Now methane production occurs by fungi, methanogens, archaebacteria, yeast cells. What is the answer? Methane production occurs by fungi, methanogens, archaebacteria, and yeast cells. Yeah, anybody answer? Anyone? Yeah. Option C. Option C. Is it correct? Ashita is saying option C. That is archaebacteria. It is correct or not? Methanogens and archaebacteria both are used in the methane production. Yeah, Akanksha is saying B. Yeah, B is also correct and C is also correct. So there will be methanogens and archaebacteria both are used in methane gas production. Okay. Now cell wall of yeast contain at high amount cellulose, chitin, muramic acid, and fructose. So simple answer is B. Venkateshwaran is saying B. That is yes. Cell wall of yeast contain chitin at high amount. Yeah, Venkateshwaran answer is good. Yes, Akanksha B is answer. Now we are moving to next slide. Now biochemical overview. Now see what is biochemical overview of fermentation. Now we know that respiration and all that stuff. Now in the respiration, many ATPs are produced, and that ATPs are used for energy purpose. All that stuff we know. So this biochemical overview discuss only about that oxidative formation and Crabtree effect. Now in this slide, the important thing is that oxidative fermentation and what is Crabtree effect. The many times question came on this, that is Crabtree effect. Now we are going to discuss only this one. Now see, fermentation reacts NADH with an endogenous organoelectron acceptor. This is a pyruvate formed from sugar through glycolysis. Sorry. Now the NADH is an important one in the fermentation which acts as an organoelectron acceptor. However, more exotic compounds can be produced by fermentation, such as different types of acids and acetone. Fermentation products contain chemical energy. Fermentation products contain chemical energy, but considered as a waste product since they cannot be metabolized further without use of oxygen. Fermentation normally occurs in an anaerobic environment. In the presence of NADH and pyruvate are used to generate ATP in respiration. This is called oxidative fermentation. That we know. What is oxidative fermentation? That we know. And it generates much more ATP than glycolysis alone. For that reason, fermentation is rarely utilized when oxygen is available. Now this last sentence line is important. However, even in the presence of abundant oxygen, some strains of yeast such as Saccharomyces cerevisiae prefer fermentation to aerobic respiration as long as there is an adequate supply of sugars. So this is known as Crabtree effect. Now what is Crabtree effect? See again. However, in the presence of abundant oxygen, some strains of yeast such as Saccharomyces cerevisiae prefer fermentation to aerobic respiration as long as there is an adequate supply of sugar, known as Crabtree effect. Some fermentation process involve obligate anaerobes which cannot tolerate oxygen. Now see, this is a simple figure that we know, that is pathway. We always say pathway that we know very well. That's aerobic respiration in cytoplasm. Now, in the aerobic respiration, glucose converted into pyruvate via glycolysis in the anaerobic condition. Then it goes for fermentation purpose where the lactic acid and ethanol is produced. When it comes inside the cell, in the aerobic process, it comes in the TCA cycle where the different types of NADH and ATPs are produced, and in another level, that's oxidative phosphorylation is there, known as electron transport chain, where the ATPs are produced. So simple pathway that we know already, which occurs in the respiration, and from this pathway we may get many types of ATPs, and we are using these ATPs as energy for our daily use. What are the questions? Now, electron acceptor in fermentation is NADH, FADH, GTP, and ATP. Yeah, A1 is correct. Venkateshwaran and Akanksha said A1. Yeah, it's correct. Electron acceptor in fermentation is NADH. Now, see, second question is very important because many times in NET, SET, and GATE the question came that Crabtree effect is. Now, I did not mention here options. I already gave here answer that we know. In the presence of abundant oxygen, Saccharomyces cerevisiae to fermentation to aerobic respiration, as long as there is an adequate supply of sugars, that we know. Now see the third question. End product of glycolysis include ATP. Now see, end product of glycolysis including ATP. What is the end product of glycolysis including ATP? First is CO2 and H2O. Second option is H2 and pyruvate. Third option NADH and pyruvate. D option CO2 and NADH. Already I gave answer, that is NADH and pyruvate is the end product of glycolysis. Now this question many times arises in NET, GATE, SET, all questions, and this year in June also this question was there. Now fourth one, which of the microbial fermentations are anaerobic? Now these also I have gave answer. Ethanol and acetone butanol, citric acid and propionic acid, penicillin and vitamin B12 and streptomycin, rifamycin. Yeah, thanks a lot. Yeah, so ethanol and acetone butanol is the microbial fermentation which is considered as an anaerobic. Now from this all stuff, we are going to move industrial fermentations. Now this is a very important topic where we are going to discuss different types of aspects within industrial fermentations. As far as industrial fermentations are considered, we have to remember different points. From that, I concluded 6 points. The first point is the cost and availability of feedstocks, means the raw material. The efficiency of industrial microorganisms. Now we have to see the efficiency of industrial important microorganisms, means that microorganism is able to produce our interested compound or not. Second thing is that that microorganism is able to produce high yield of that compound or not. That we have to see is nothing but the efficiency. Fermentation condition and optimization. Now we know that if we want to produce a good compound or high yielded compound, that time we have to be with fermentation conditions and optimize the conditions. Then downstream process and end product recovery efficiency. Now downstream process is very important topic as far as fermentation is considered, because what actually happens during the production of any product, we can produce it by using the fermented media and the microbes, but the downstream process is very critical because there are different types of process. For example, filtration is there, chromatography is there, electrophoresis is there, precipitation is there. So which process suitable for our product, that we have to decide. And the end product recovery efficiency. Now fermentation byproduct utilization. Now see, during the fermentation, not only our product get produced, there are different byproducts are also there. There are different byproducts. So how we can escape ourselves or that product from that byproduct, we have to discuss that. Next one, utility, consumption and labor cost. Now utility, consumption and labor cost we have to see, because as far as industrial product is considered, at the end question arises that end product and end yield of that product, and end yield is considered, then we have to see the cost of that product in market. As far as this cost of product in market, as far as the yield of that product we considered, we have to consider labor cost and utility consumption. Now this is one of the important topic as far as industrial fermentation is considered, that is the design of fermenter. Now you can see the figure. Now in this figure, known as design of fermenter. Now this design of fermenter is made up from different types of materials. Now see, this is a motor. At the top there is a motor. Now motor is at the bottom of the fermenter. Now there is a sterilized nutrient solution inoculated with a pure culture of the microorganisms. Means this is the inlet of fermenter from where we can add the microbial cultures. Now there is a sampling tube, means we can remove the samples. Now here then impellers are there. Impellers are also called as stirrers. The impellers are used for the mixing of the microorganisms and the nutrients and distributing the heat. Now you have to remember the impellers function. Now what is the impellers function? Mixing of the microorganisms and nutrients and distributing the heat all over the fermenter. Now next one is cooling water in. Now during the fermentation there is generation of heat. So for that there is a cold water requirement. So that cold water is provided by this well. Now below this there is sparger for forcing air into the medium. Now sparger is required for the aeration purpose. Now in this left side, we saw first inlet for the inoculation of microorganism, then sampling tube, then impellers. Now we have to remember that what is the function of impeller. So mixing the microorganisms and nutrients and the distributing the heat. Now there is a cooling water inlet. That is an inlet, is it? Yeah, yeah, it's inlet only. Yeah, Venkateshwaran, it's an inlet only. Now sparger for forcing air into the medium. Now sparger is used for aeration purpose. Okay, now we are moving right side. Now right side, temperature and pH monitor is there. Then there is exhaust. Then a stainless steel vat is nothing but the fermenter of fermenter's style, that is stainless steel fermenter is used where the microbes became trapped and corrosion of the surface. This is the upper layer or top layer of fermenter where the space is present. Now this space, we know that in each and every fermentation procedure we are using different types of fermenters. Now at the top of this fermenter we are keeping some space for the aeration purpose or for the trapping the microorganisms or to avoid bubbling also. Then below this there is a cooling water out. Then below this water jacket for circulating cooling water to remove the heat generated by the metabolic activity of the microorganisms. And at the end there is sterile air inlet and there is a products in outlet. So this is the design of fermenter. From this, what you have to remember, first thing is that impellers function. Okay, next thing is that sparger function, and why we have to keep space at the top of fermenter, that you have to remember. Now the different substrates used in the fermentation procedures. Now these are the different substrates. First is sugarcane bagasse. Sugarcane bagasse is majorly used in the alcoholic fermentation. Now tea waste. Wheat bran. Now majorly wheat bran is used in different types of organic acids production and vitamins production also. Now sawdust is there, apple pomace is there, and coconut oil cake is also there. Now these are the majorly used substrate in the fermentation procedures. Now the questions. Okay, first question is that mixing of microorganisms is carried out by, now first sparger, impellers, oils and acids. Now simple answer is impellers. You are getting no? Everyone? Yes, Akanksha. Yes. So mixing of microorganisms is carried out by sparger, impellers, or oils or acids. So simple answer is by impellers. Second question, sparger is used for which purpose? So simple answer is aeration. Now sparger is used for aeration purpose. Now in wine production or fermentation, which type of fermenter is used? Yeah, Venkateshwaran, I already labeled as a red, that we can see the answer as answer. So in wine fermentation, wood fermenter is used, not steel fermenter. Water jackets are used in fermenter for aeration, mixing of microorganisms, cooling of fermenter, inoculation. So simple answer is cooling of fermenter. The last one, dash dash used in alcohol fermentation. Molasses, yeast cell, Saccharomyces cerevisiae, and all. What is the answer? Yeah, Venkatesh is saying is D. Yeah, it's correct. Is D, because in alcohol fermentation we use molasses as a substrate, yeast cell, and Saccharomyces cerevisiae is nothing but only yeast cell. So answer is all. Yeah, we are going to discuss in further why food fermenters are used in the wine fermentation, that we are going to discuss afterwards. Okay, thanks a lot for question. Next slide. Now types of fermentation. Now if you see the types of fermentation, there are main three types of fermentation: batch process, then fed batch fermentation, and continuous fermentation. Now see the first, what is a batch fermentation procedure? Now as far as batch fermentation is or batch process is considered, where only one batch is used for the production of that product. For example, in lab, what we are doing in lab. So in lab, what we are doing, sorry, simply we are taking one flask, and in that we are adding our material or media with microorganisms, and we are keeping it for incubation purpose. After incubation, we are getting that product, and we are doing filtration, then solvent extraction, then chromatography and all that. Then we are getting our product. So batch means what? We are doing fermentation at once by using one time microorganism and by using one time substrate or raw material or media. We are not adding again and again media and microorganisms in that fermenter. We are adding everything once, and after addition of all this stuff, we are doing fermentation, known as batch process, where it occurs in batches only. It is not continuous. It is in batches only. However, it can be expensive because of the fermenter must be sterilized. It's still in question slide. It's still in question slide. Venkateshan, what do you want to say? Now that why food fermenter for wine production is used, that we are going to discuss. Afterwards. Okay. You are seeing the types of fermentations. Can you see this slide? Hey all, can you see the types of fermentation slide? Anybody answer me? Yeah, yes. Now this is the batch process what we discussed now. The batch fermentation goes through a series of phases. There is a lag phase in which cells adjust to their environment. Then a phase in which exponential growth occurs. Once many of the nutrients have been consumed, the growth slows and becomes non-exponential. But the production of secondary metabolites accelerates. This continues through a stationary phase after most of the nutrients have been consumed and then the cells die. Now we know that batch process. What is batch process? So production of that compound occurs in batches only, not continuous, known as batch process. Where during this batch process, the microorganism goes through different types of phases: lag phase, log phase, stationary phase, and decline phase. Next one is fed batch fermentation. Now in the fed batch fermentation, what is happening? Now see. Now it is a variation of batch fermentation where some ingredients are added during the fermentation. This allows greater continuation the stages of the process. In particular, production of secondary metabolites be increased by adding a limited quantity of nutrients during this exponential growth phase. Fed batch operations are often sandwiched the batch operations. Now fed batch fermentation is nothing but the addition of nutrients, addition of microorganism during the fermentation. During the fermentation is known as fed batch fermentation. While in the batch fermentation, we are not doing the same thing. We are adding fermented media and microorganism once at the start of fermentation. While during the fermentation, we are not adding anything in the batch fermentation. While in fed batch fermentation, we are adding microorganisms and the ingredients also. Now in the continuous fermentation, we are adding microorganisms and the ingredients during the fermentation. At the same time, we are extracting our product also. So there are different types of continuous fermentations: chemostat, turbidostat. These are majorly known, chemostat and turbidostat, where the continuous fermentation is carried out. Now see, these are the three types of fermentations: batch fermentation, continuous fermentation, and fed batch fermentation. Now you can see the diagrams. Now from this diagram we can understood the batch fermentation is only one flask is there. Okay, only one fermenter is there, where there is no addition. Now this at the top of this, okay, sorry, at the top of this fermenter you can see these lines. It indicates that addition of nutrients and all that. Now in batch there is no any line at the top, so it indicates that there is no addition of microorganisms and ingredients during the fermentation. While in the fed batch, in the third diagram, you can see there is a one line at the top. It indicates that there is addition of microorganisms and ingredients during the fermentation in the fed batch. Now in the continuous, you can see the two lines at the two sides. First is at the left and second one is at the right. It indicates that addition of microorganisms and addition of ingredients, while second one indicates that is the right one, removal of product. So this is all about the batch, continuous, and fed batch fermentation. You have any questions about this? Any questions about batch, continuous, fed batch fermentation? Okay, no. Thanks a lot, Sorna Chaudhary, ma'am. Okay, no. Now see, this is the microbial growth. Now this is a very important aspect as far as fermentation is considered. Okay, thanks, ma'am. As far as microbial growth is considered. Now see this graph. In this graph, you can see I'm not able to highlight it. Sorry for that, because I'm doing first time. And you can see this graph. In this graph, you can see the concentration against time. Thanks a lot, ma'am. Now in this, you can see the concentration against time. Now see, this is the green one. It's showing limiting nutrients up to this middle line. Afterwards, it's growing and it's showing secondary metabolite. Now here, the first one, black, it's growing from bottom and going top, which indicates cell mass. Okay, and at the top they indicates primary metabolism in trophophase and secondary metabolism in the idiophase. Now we have to understand this graph first. Now see the cell mass. Now see the cell mass and the limited nutrients. Now see the cell mass in the trophophase. Now see the first trophophase. Now in the trophophase, cell mass is very less. It's very less, while nutrients are more in amount. While cell mass is increasing in trophophase, while at the same time nutrients are decreasing in the trophophase. But at the same time, primary metabolites are producing by the microorganisms. While when cell mass comes in idiophase, at the same time, limited nutrients are there, but cell mass is increased and secondary metabolites are produced at high amount. So from this graph, what we can understood, so in the trophophase, there is a high production of primary metabolites. In the idiophase, there is a high production of secondary metabolites. While in the trophophase, limited nutrients, means nutrients declines in the trophophase, while in the idiophase, secondary metabolites produced at a high amount. Now, you have to remember from this graph, as far as competitive exam is considered, secondary metabolites are produced in which phase? This is a very important question. This question arises in many times in NET GATE set questions, exams, that secondary metabolites are produced in which phase? So simple answer is idiophase. Okay, now see the questions. The first question is, yeah, secondary metabolites are produced in which phase of microbial growth? Trophophase, idiophase, both, none of the above. So simple answer is, what is answer? Yes. What is answer? Idiophase. Yes, B is the answer, idiophase. Second, bread and alcoholic beverages are produced by. This question arises in need. Yeah, it is mentioned. Thanks a lot, ma'am. Bread and alcoholic beverages are produced by which fermentation procedures? Batch, fed batch, and continuous. So answer is batch fermentation. Cell biomass increases in which phase of microbial growth? So from graph we can say that cell biomass increases in idiophase of microbial growth. Primary metabolites are produced in which phase of microbial growth? So in trophophase, primary metabolites are produced. Now chemostat is an example of which fermentation process? So chemostat is an example of continuous fermentation process. Now alcoholic fermentation. Now see the alcoholic fermentation. Now this is very important aspect as far as fermentation is considered. Now in alcoholic fermentation, sugarcane, corn, sugar beets are used as a substrate with addition of gasoline also. And for the alcoholic fermentation, majority yeast cell is used known as Saccharomyces cerevisiae. Now we saw in the before slides. Now see this figure. Now glucose is converted into pyruvate. ATP generation is there. Now this pyruvate get converted to 2-acetaldehyde. This 2-acetaldehyde get converted to ethanol. Okay, ethanol. Now this is the simple alcoholic fermentation where the alcohol fermentation occurs. The acetaldehyde is reduced into the ethanol using the energy and hydrogen from NADH, and the NADH is oxidized into NAD+, so that the cycle may repeat. The reaction is catalyzed by the enzymes pyruvate decarboxylase and alcohol dehydrogenase. Now these enzymes you have to remember, pyruvate decarboxylase and alcohol dehydrogenase. These enzymes are very much important as far as competitive examinations are considered. Now lactic acid fermentation. Now see, the lactic acid fermentation is shortly known as LAF, that is LAF, is carried out by the Lactobacillus streptococcus leukinostoc bacteria, and it is also carried out by the fungi, algae, and protozoa. Here pyruvate acts as an electron acceptor. Now you have to remember the microbial names, that lactic acid fermentation is carried out by the Lactobacillus streptococcus leukinostoc bacteria. Now there are two types of fermentation procedures of lactic acid. First one is homolactic and second one is heterolactic. Now first understand what is homolactic. Now homolactic fermentation means what? There is a production of only lactic acid. There is no any byproduct. That is why this fermentation is known as homolactic fermentation. Well, heterolactic fermentation means what? There is a production of lactic acid as well as there is production of other byproducts. That is why this is known as heterolactic fermentation. Now see, there are some reasons to go further, or there are some reasons for the further decomposition or further production of different types of byproducts in the heterolactic fermentation. Now what are these reasons? Now the acidity of lactic acid impedes biological processes. This can be behavior the fermenting organism as it drives out competitors that are unadapted acidity. As a result, the food will have a longer shelf life. However, beyond a point, the acidity starts affecting the organism that produces it. The high concentration of lactic acid drives the equilibrium backwards, known as Chartier's principle, decreasing the rate at which fermentation can occur and slowing down growth. Ethanol, into which lactic acid can be easily converted, is volatile and will readily escape. allowing the reaction to proceed easily. CO2 is also produced there, but it is only weakly acidic and even more volatile than ethanol. If propionate, butyrate, and longer monocarboxylate acids are produced, the amount of acidity produced per glucose consumed will decrease as with ethanol, allowing the faster growth. Any doubts in this slide? Any doubts in these slides? Okay. Okay. So next one, mixed acid fermentation. So mixed acid fermentation, where the hydrogen gas is produced in many types of fermentation. Okay, the majority known as mixed acid fermentation. Now the butyric acid fermentation, caprolate fermentation, glyoxylate fermentation, etc. as a way of regenerating NADH. Electrons are transferred to ferredoxin, which in turn is by dehydrogenase producing. Okay, so hydrogen gas is a substrate for methanogens and sulfate reducers, which keep a concentration of hydrogen low and favor the production of such as an energy compound. But hydrogen gas at a fairly high concentration can nevertheless be formed as in flat earth. An example of mixed acid fermentation, bacteria such as Clostridium pasteurinatum ferment glucose producing butyrate, acetate, carbon dioxide and hydrogen gas. And this is the reaction which leads to a production of acetic acid. Glucose could theoretically be converted into just CO2 and H2, but the global reaction releases little energy. Now from this slide, what we have to remember that the microbial name, that is Clostridium pasteurinatum. Okay, the Clostridium pasteurinatum we have to remember, which is used in the mixed acid fermentation. Now, antibiotics fermentation. Now, as far as antibiotics fermentation is considered, there are different types of antibiotics which are produced at industrial level, but the conditions are differ from antibiotic to antibiotic because of their maintaining structure, because of the microorganisms used in that fermentations. Now, first, that is media preparation. Now, media usually contain its carbon source, which is found in corn strip liquid and glucose. Now then heat sterilization is there. Then after media preparation we have to do sterilization. Then directly fermentation we have to do by the addition of microorganisms. Okay, then biomass removal is there. By using rota vacuum filter we can remove the biomass using the phosphate buffer. Then we have to do solvent extraction. Then after solvent extraction we have to do centrifugal extraction to separate the solid waste from the liquid solution containing penicillin. Now this is for penicillin only. And last one, there is a drying. Drying is necessary to completely remove the moisture contained from the penicillin salt. Now this is the classification of antibiotics. Now based on the action, bacteriostatic and bactericidal, and based on the spectrum of action, broad spectrum and narrow spectrum. Now this is the some list of antibiotics produced from the microorganisms. Can you see the list? Yeah, I know that it's somewhat zigzag, but it's most important. Now see the antibacterial. Now penicillin G is produced by penicillin species, cephalosporin, acremonium species. You can note down if you want, okay. Then streptomycin is by streptomyces species, tetracycline, streptomyces, chloramphenicol, cephalosporin, bacitracin is also by bacillus species. Now from this, streptomycin is most important, which is produced by the streptomyces species. Location arises on that, and tetracycline also. Anti-tumor, actinomycin D, streptomyces, mitomycin, streptomyces, bleomycin, streptomyces, erythromycin, streptomyces, and leomycin, streptomyces. Antifungal, disofulvin is there, the penicillium species. Now food preservative, natamycin, streptomyces species, and nicin also by streptomycin species. Antiprotozoal, adenorubicin, streptomyces species. Antituberculosis, rifamycin is there, produced by Nocardia species. Antiemobic is there, tetracycline, streptomyces species, and Fumagili aspergillus species. Amino acid production. Now, in the amino acid production, I choose the important one which are in syllabus. Now, first one, glutamic acid. Now, natural products such as sugarcane is used as a raw material for the glutamic acid fermentation. Then the sugarcane is squeezed to make molasses. The heat sterilized raw material and other nutrients are put in the tank of the fermenter. The microorganisms, Corynebacterium glutamicum, producing glutamic acid is added to the fermentation broth. The microorganism react with sugar to produce glutamic acid. Then the fermentation broth is acidified and the glutamic acid is crystallized. Now see the fermentation. Now for the glutamic acid fermentation, molasses is used as a raw material, and the microorganism Corynebacterium glutamicum. You have to note down this. This Corynebacterium glutamicum is used in the glutamic acid fermentation, where this bacteria produces glutamic acid from the raw material. And at last, the fermentation broth is acidified and the glutamic acid is crystallized. So glutamic acid is found in crystallized format. Next one, L-lysine production. Now this is one of the important production fermentation process. Now essential amino acids for human nutrition, used as supplement for bread, cereals, and other food. There are two steps for the production of lysine. First one is the formation of DAP, that is nothing but the diampinopidic acid, which is formed by E. coli. Next one, decarbosylation of DAP by DAP decarboxylase enzyme from Enterobacter aerogenes. Now you can see the reaction where the DAP converts into lysine in the presence of Escherichia coli and Enterobacter aerogenes. Now in this slide, what you have to remember. So first one, the conversion of or the formation of DAP, bacteria Escherichia coli, and the decarbosylation of DAP by DAP decarboxylase enzyme. Bacteria name is Enterobacter aerogenes. That this you have to remember. Now there are some important fermentation products. First one, ethanol, Saccharomyces cerevisiae. Organism is used and it is used industrial it is used as solvents, beverages purpose. Next one, glycerol. Organism is used as Saccharomyces cerevisiae. You have to note down this if you want. Okay, this is very important. Glycerol or glycerol is produced by the Saccharomyces cerevisiae. Use production of explosives. Then lactic acid, organism Lactobacillus bulgaricus. Use food and pharmaceutical. Acetone and butanol, Clostridium acetobutylicum. Solvents, alpha amylase, Bacillus subtilis, starch hydrolysis. Now if you have to note down this, my organisms used in the product, I can say for some time. Now enzyme production. Now enzyme production is one of the important factor as far as microbial fermentation at industrial level considered. Now see, the amylase enzyme is produced by the microbe Aspergillus oryzae, Aspergillus niger, and Bacillus subtilis, and used for the production of alcohol and all that. Cellulose is produced by the Aspergillus niger, used for alcohol and glucose production. Now the invertase is used for the sucrose inversion and in confectioners and produced by the Saccharomyces cerevisiae. Pectinase enzyme produced by the Aspergillus species and used for the clarification of fruit juices, alcohol production. Glucose oxidase is used for the antioxidant in prepared foods and produced by the Aspergillus niger. Note down this if you want. Any doubts in this slide? Okay, no doubts. Thanks a lot. Now vitamin B12 production. Now see, In the vitamin B12 production, raw material is used soybean meal, yeast, and casein with cobalt. Now, cobalt is one of the important factor in the vitamin B12 production. That question was arised that which is one of the important factor used in the vitamin B12. So, cobalt. Microorganisms used Streptomyces griseus, Streptomyces olivaceus, Bacillus megaterium, Pseudomonas denitrificans, Propionobacterium, Ricky and Proteus species. Now these are the important microorganisms used in the vitamin B12 production. Production media contains distillers soluble 4%, dextrose 1%, CaCO3 0.5%, and COCl12 6H2O 1.5%. Fermentation conditions: temperature 80 degree Fahrenheit, pH 5, aeration 0.5 volume medium per minute. Antifoam agents, corn oil or lard oil or silicones are used. Yield 1 to 2 milligram per liter of fermented broth. Recovery is carried out by filtration, then solvent extraction, the rotaevaporator, and dried powder. Then treat with cyanide and store it or use it. Now in this slide you have to remember the raw material used in the vitamin B12 production and the microorganisms used in the vitamin B12 production and the fermentation conditions. Now vitamin B2 production. Here raw material is same, soybean meal, yeast and casein. Microorganisms are used Rheomethicium ashby and ashby gossypi. This is very important question, point. Microorganisms used in vitamin B12 production is Rheomethicium ashby and ashby gossypi. Production media contains glucose, collagen, sucrose, glycine, and soya oil. Fermentation conditions are same as like previous. The recovery is by heating of fermented broth at 120 degrees Celsius for 1 hour. After that, we have to do filtration. Then after that, with filtrate, we have to do precipitation by using dithonite, and at last we have to do crystallization. Then note down the microorganisms used in the vitamin B2 production and the recovery process, that precipitation by dithonite and crystallization. The vaccine production. Hepatitis B is one of the important and majority questions asked on this only. That's why I took this. Now vaccine production in this hepatitis B is produced by recombinant DNA technology, where the segment of viral DNA which is able to express hepatitis B surface antigen is cloned into Saccharomyces cerevisiae, and that Saccharomyces cerevisiae is used in the fermentation. Now, production media is yeast extract, soya, peptone, dextrose, amino acids, and mineral salts. Fermentation conditions as like this, but the recovery is somewhat different for the vaccine production. Now see, breakdown of yeast cell. After fermentation, we have to do breakdown of yeast cell. Then physiochemical steps for purification. Purified protein treated with formaldehyde and then co-precipitate with the alum to form bulk vaccine. Then we have to use adjuvants, and then vaccine will be purified and store it. Now the questions. Important enzymes in alcoholic fermentations. Already I gave this answer. No options are there. Pyruvate decarboxylase and alcohol dehydrogenase. These are the two important enzymes in alcoholic fermentation. Now lactic acid bacteria are Lactobacillus, Streptococcus, Leconistic bacteria. Now what is homolactic fermentation? So where only lactic acid is produced. What is heterolactic fermentation? Where rather than lactic acid, other products are also produced. Now mixed acid fermentation is carried out by Clostridium paschareum. Penicillin is produced by Penicillium chrysogenum. Rifamycin is produced by Nocardia species. Now glutamic acid is produced by Corynebacterium glutamicum. L-lysine is produced by Escherichia coli and Enterobacteria aeruginosa. Corn strip liquid is the substrate for L-lysine production. Amylase is used for Aspergillus niger. Invertase is produced by Saccharomyces cerevisiae. Vitamin B12 is produced by Streptomyces griseus, L. oleaceans, Bacillus megaterium, Pseudomonas denitrificans, Propionobacter species. Basic and important substrate for vitamin B12 production is cobalt. You have to remember this. And vitamin B12 is produced by Rheomethicium ashby and ashby gossypi. And hepatitis B is a rDNA vaccine. So thanks for your attention. Hello, sir. Ah, sir. Yes, it's going on. Yeah. It's over, sir. Now questions are going. Okay, okay, sir. Yeah. Okay. Ah, yeah, okay. Okay. Okay, thanks a lot, ma'am. Pleasure to see here. Suvarna, ma'am. Any questions? Yeah, my email ID. Okay, thanks a lot. My email ID as in first slide I mentioned. Wait. Yeah, this is my email ID rohitmane2025@gmail.com. Yeah, Venkateshwaran, this is my email ID rohitmane2025@gmail.com. Yeah, any questions, any doubts? You can ask me on email or you can take my mobile number also 8975957736. 8975957736. Okay, thanks a lot, guys.